Hello everyone, my name is Shivani Kumari, working as an assistant professor in the Department of Pharmacy at RPIIT campus. Today, I will explain some topics of biotechnology 6th sem like transduction, transformation and conjugation. Transformation is a process where certain competent bacteria are able to take up free DNA released by other bacteria. Transformation is a kind of genetic recombination which involves the passage of only gene carriers. Competence in certain bacteria is governed by certain proteins which include membrane associated DNA binding protein, cell wall, autolysin, and various nucleases. The mechanism involved in transformation has been described. Transformation takes place in a few cells of mixed population. A few donor cells break apart, resulting in an explosive release and fragmentation of DNA. A fragment of double-stranded DNA, 10 to 20 genes, get attached to the recipient cell for entry into the recipient cell. During entry, one strand of the fragment is dissolved by the enzyme, thus only the second strand is left, which passes into the recipient cells through cell wall and cell membrane. Within the recipient cells, a portion of single strand of double-stranded DNA of recipient cell is displayed by the enzyme and then displayed by the DNA of donor cell. The displaced DNA is dissolved by another enzyme. Thus, the recipient cell becomes transformed, displaying the characteristics of its own as well as of the newly incorporated DNA. Second one is the transduction. Transduction is a genetic recombination which involves transfer of genetic material from the donor of the recipient cell through a non-replicating bacteriophage. The transduction is divided into two parts: generalized transduction and specialized transduction. In generalized transduction occurs more commonly than the specialized transduction. The prophase particle is present in the cytoplasm of the infected bacterial cell. The phage DNA starts synthesizing new phages. The chromosome of bacterial cell get fragmented. Some fragments get attached to the DNA of some new phage particles while the other remain with phage DNA. The newly formed phage with fragment of bacterial chromosome in its DNA attack a new bacterium. As a result, the gene of the parent bacterium is transferred to the new bacterium and cause recombination. Second one is the specialized transduction. The bacteriophage gets attached to a bacterial cell wall at the receptor site and its nucleic acid is transferred into the cytoplasm of the host cell. The phage does not cause lysis of the host bacterium. The phage nucleic acid codes for the synthesis of repressor protein in the bacterial cell. These proteins prevent the virus from producing the material required for its The viral DNA exists either as a fragment in the cytoplasm or attach itself to the chromosome known as prophase. The bacterial cell which carries the prophase is lysogenic, and the phenomenon in which the phage DNA and bacterium exist together is termed lysogeny. In conjugation, two cells of opposite mating type F positive and F negative attach with each other by sex pillars. There is a hole of 2.5 micrometer diameter in sex pillars through which DNA can pass from the donor to the recipient cell. The F factor or F plasmid is a double-stranded DNA loop found in the cytoplasm along with the nucleotide. After the conjugation tube established, the F factor prepare for replication by the rolling circular mechanism. The two strands of F factor separate from each other and one of them passes to the F negative cell called as recipient cell. On reaching the F negative cell, the enzyme synthesizes a complementary strand to form a double helix. In F positive cell, called donor cell, also a new DNA strand is formed to complement the leftover DNA strand of the effector. Thank you.